Well, I was talking about a cell death pathway called apoptosis. It turns out that when cancer cells are killed by chemotherapy, they don't just sort of fall apart and die in some nonspecific way, but there's actually a program of cell death that is triggered, and the cells actually are forced to commit suicide. Some cancer cells are able to escape this, and I'm particularly interested in understanding how they escape and how we can target that to kill them. I myself am not a clinical trialist, but there are a couple clinical trials we've proposed based on the idea of antagonizing BCL2, which is an anti-apoptosis protein. And hopefully we'll be able to start those clinical trials in the next year.
And what ideas were you putting forward about apoptosis in your talk just now?
Well, I was talking about breast cancer, and it looks like, at least in some cases, when breast cancer cells become resistant to Taxol, that they do it by means of making a lot of an anti-death protein, an anti-apoptotic protein called BCL2 or BCL-X-long. And there is now, made by Abbott Laboratories, a very good BCL2 antagonist known as ABT263, which is in clinical trials in phase 1. And what we've suggested is a clinical trial combining this BCL2 inhibitor with Taxol in patients who are Taxol resistant, because we found in the lab that if we take cells that have acquired a resistance to Taxol, we can restore sensitivity by co-treatment with these BCL2 antagonists.